Let's see if you can figure out why Gene has never met a female that wasn't a double crosser. And I'll give you a hint, it's not because Gene is just an angry guy. So by the end of this PowerPoint, you should be able to use a Punnett squared to predict an offspring's genotype and phenotype for sex-linked traits, and also compare and contrast how traits on autosomes and sex chromosomes are inherited. So in all of your body cells, you have 46 chromosomes. 44 of them are considered autosomes, and those are pairs 1 through 22. The sex chromosomes come right after that, the X and the Y. Sex chromosomes determine whether the person is a male or a female. And so this particular individual is a male because they have an X and a Y. A female would have two X's. So on these sex chromosomes are some surprising genes. Even though the X chromosome is considered a sex chromosome, it doesn't have any genes that have things to do with gender on them. It has genes on there like color blindness, hemophilia, Duchenne muscular dystrophy, and that's just a disease where muscles progressively become weaker. So the X chromosome doesn't have any genes on it that have to do with gender, but the Y chromosome does. Only males have the Y chromosome, and so the Y chromosome contains all of the genes that are needed to make a boy a boy. It's the genes on the X chromosome, though, that are considered sex-linked genes or sex-linked traits. And they behave a little bit differently than traits on the autosomes, mainly because men have one and women have two X chromosomes. So they have to behave the same way. So women's X chromosomes do something a little bit strangely. Also, when you do a Punnett square with sex-linked traits, you need to use the X's and Y's to show that these are sex-linked traits. So the X and the Y represent the sex chromosomes of the male parent, and the X and X represent the sex chromosomes of the female parent. Hemophilia is a sex-linked trait that is recessive. So what that means is that it can get masked if there's a dominant trait. So in this female, the female has the dominant trait, which isn't shown. That's just the way that it gets written. The dominant trait which isn't shown, which just means that that X chromosome carries a normal allele, so that she wouldn't have hemophilia. And then her other X chromosome carries the hemophilia allele. And since hemophilia is recessive, a female needs two copies of that recessive allele in order to have hemophilia. This female has a dominant trait, so that dominant trait could mask the recessive hemophilia trait. So this female does not have hemophilia. She's what's considered a carrier. She's considered a carrier because she's able to pass hemophilia down to her children, even though she does not have hemophilia herself. The male, however, so the X chromosome is the only place that you find hemophilia. Hemophilia is not on the Y chromosome. So a male only has one X chromosome. So if the male gets an allele for hemophilia, then the male has hemophilia. He doesn't have another X chromosome that has the dominant trait which can mask his hemophilia. So a male needs one X chromosome with a particular trait in order to have that trait, and a female needs two in order to have that trait. So let's take a look at the Punnett square. So we write the sex chromosomes along the top and the side to show the female parent and the male parent, and we include, so this little H is showing the allele for hemophilia. So we're crossing here a woman who's a carrier because she only has one. She doesn't have hemophilia, but she could pass it on. And we're crossing her with a male who does have hemophilia. So we're going to fill out the Punnett square just like normal. We'll bring across letters on the side and bring down letters that are on the top. And then if we look at the possible offspring that these people can have, this is a female who's a carrier of hemophilia. This is a female who actually has hemophilia. This male is normal. He does not have hemophilia. And this male has hemophilia. So when we write out the genotype and phenotype ratios, we want to include that males and females are different. So we can't just say like that 50% of their children have hemophilia. We need to be specific about males versus females. So the genotype ratio, remember, are just the letters. So we write out the genotype ratio, and there's a 25% chance of having each of these genotypes. And then with the phenotype ratio, or what these people actually look like, or diseases that they actually have, we need to specify each one individually because each of these offspring are different. So here we've got a female who's a carrier. That's a 1 out of 4, or 25% chance of having a female who's a carrier for hemophilia. This female has hemophilia because she's got 2 little h's, no big H or absence of a little h to mask it. So she has hemophilia, 25% chance of a female with hemophilia. And then you've got here a male who's normal or does not have hemophilia. And here's a male who does have hemophilia. Even though he just has 1 H, he doesn't have another X to mask this H. So there's a 25% chance of having a male with hemophilia. So now you try. Cross a woman who has hemophilia and a man who does not. Write the genotype and phenotype ratios for their offspring. So pause and do that now, because I'll show you the answer in just a minute. So here's the Punnett square that you should have come up with. We've got the female along the top who has hemophilia, so she must have 2 little h alleles. And the male who does not have hemophilia, so along the side he has no little h's. And so we bring across the letters and we bring down the letters, and we get 2 different genotypes. Either we get a male with the little h, or we get a female with 1 little h. And so what that translates to in terms of phenotype is that all of the males have hemophilia because they've got a little h, and all of the females are carriers of hemophilia. They have 1 little h as well. So the X chromosome behaves a little bit differently in females than it does in males because the body can really only deal with having 1 X chromosome. Males have 1 X chromosome, so their X chromosome doesn't need to do anything special. But because females have 2, one of them gets what's called inactivated. So when it gets inactivated, that chromosome gets really dense. It crumbles up into even a smaller ball. It's sort of like if the message that's contained in DNA is sort of like words written on a page. If you crumble up that page, you can't read the words anymore. Well, a bar body or an inactivated X chromosome is sort of like you've crumbled up that piece of paper. You can't read anything that's on there anymore, and it's essentially useless. So in each one of a female's cells, she has 2 X chromosomes, and 1 gets converted to a bar body as she's developing in her mother's womb. So the way that they get converted to bar bodies is totally random. So here we're showing the 2 X chromosomes inside of each one of a female's cells, and one of them randomly has turned into a bar body, or this crumpled up chromosome that doesn't get used. And so one cell might be next to a cell that has the opposite chromosome. And so what that can produce is sort of like a mosaic effect. So there's patches of cells that are showing the gene for one of the X chromosomes, and right next to cells that are showing the gene for the other X chromosome. It just depends on which one of the X chromosomes gets converted into a bar body. So because females need 2 copies of X-linked or sex-linked traits, men have a lot more sex-linked diseases than females do because they only need one copy of that trait in order to show it. So colorblindness and hemophilia and Duchenne muscular dystrophy are examples of diseases that affect men more than they affect women, just because men need one copy and females need two. So let's see if you can figure out what's going on with this cat. There's a certain species of cat where males can either be orange or they can be black. And I guess I should say they're back because they generally have a white underside. So the tops of them can either be orange or they can be black. But what's going on with this cat that has patches of black and patches of orange? What gender do you think this cat is, and how did it get black and orange patches?